Welcome back to our last Forensics 306 lecture of the year. Congratulations on making it this far, you guys. Today, on a heavier note, I'm sure you guys have all heard of the murder that just happened on UBCO Okanagan's campus. This murder happened in the laboratory around 5 o'clock Saturday last. As of right now, there are 4 suspects that were seen leaving the laboratory around the same time as this murder was thought to happen. I have been made the lead investigator on this case as this is my area of expertise. So I was thinking this is a unique opportunity as I go through and analyze the data that was collected from the crime scene to share this with you and let you all get a first-hand experience of what happens in the field and what you guys will be doing once you have graduated. There have been 2 main pieces of evidence collected from the crime scene. There was some dried droplets of blood collected from near the victim, and there was also some dried skin cells collected off the murder weapon. Both of these samples were determined to not be of the victim, and therefore they must be of one of the 4 suspects. Being the lead investigator on this case, it is also important that I analyze my own DNA to rule out myself as a suspect. Also, more importantly, to use as a control in case my DNA contaminated anything during evidence removal or something like that. So I will be taking a swab of my cheek and giving my DNA for analysis as well. So I'm sure you guys remember talking about DNA profiling earlier in the term. We talked about many different ways to analyze DNA and compare suspect DNA to crime scene DNA, but I'm going to briefly touch on the 3 main types of analysis techniques that we are going to use in this investigation. And if you guys are confused on any of them, please refer back to your earlier notes as we have went highly in depth on these techniques earlier in the term. First off, remember earlier in the term when we learned about DNA profiling, we learned that it was important to analyze the polymorphic parts of DNA. This is because the polymorphic parts of DNA are the parts of the genome that are different between individuals, and this is actually only about 1% of the entire human genome. Approximately 99% of our genes are genetically identical, but the 1% that differs is very useful in determining individuals from one another. Typically within these polymorphic sequences, the anonymous regions are analyzed. This is because the anonymous regions are derived from parts of DNA that don't code for any known proteins or traits, and therefore they don't give out any information about the individual, which helps keep everything confidential and conserve the identity of the suspects during analysis. From the evidence collected at the crime scene, there are going to be 3 different types of tests performed to help determine which suspect committed the crime. The first test performed will be restriction fragment length polymorphism analysis, also referred to as RFLP analysis. To perform this analysis, you have to have a lot of high quality DNA. So this will be performed on the drops of blood collected near the victim because this was determined to be enough DNA and of very high quality. In RFLP analysis, the sample DNA is cut with various restriction enzymes. Remember, these restriction enzymes recognize specific sites within the DNA and cut at a specified location, resulting in different sizes of fragmented DNA. This DNA is then ran on a gel, allowing the DNA to separate into different size fragments. The pattern of all the fragments that are created from a given person is referred to as a genetic fingerprint and varies between each individual because everyone's DNA is slightly different with respect to where these restriction enzyme sites are created, which means when the DNA is cut with these restriction enzymes, the fragments that are resulting from this will be different in each individual. The different size fragments can then be visualized on the gel and compared to one another, and the suspect fingerprints can also be compared to that of the DNA that was collected and analyzed at the crime scene. When you look at the resulting gel, it should be very clear that the pattern of bands seen from a certain suspect will directly match the pattern of bands seen from the crime scene DNA. One benefit to using RFLP analysis is you do not need to perform a PCR reaction beforehand, but a limitation is that you need a large quantity of high-quality DNA for this analysis to be accurate. The next test that is going to be performed is short tandem repeat analysis. This is also just referred to as STR analysis. This can be performed on smaller amounts of DNA and also can be performed on DNA of questionable quality. And that's because the short tandem repeats are repeats of only 2 to 9 base pairs long, meaning partially degraded DNA or a small amount of DNA would typically still have an intact short tandem repeat sequence. The number of STR repeats found within an individual varies between the population, and the number of repeats is inherited in a Mendelian inheritance pattern. That means the number of repeats on one chromosome is inherited paternally, while the number of repeats on the other chromosome is inherited maternally. This allows an individual to either be heterozygous or homozygous for the number of repeats at a specific locus. The TH01 locus is going to be used for STR analysis. To increase your accuracy and increase the ability to rule out suspects, it would be beneficial to use more than one locus. However, we are just going to use one for this analysis because there is a very low number of suspects. What we're going to do is we're going to look at the number of alleles that are seen at the TH01 locus, and we're going to compare the DNA from all the suspects to the DNA found at the crime scene. PCR will be used first to amplify the repeated sequence to make sure there is enough DNA to accurately compare individuals. I'll tell you guys a little bit more about PCR in a few minutes. I think we've touched on it briefly before in the term, but not very in depth, so I will be going through that in a minute. Just hold tight. STR analysis is very beneficial because it can be performed on low-quality DNA. However, a limitation is that DNA must first be amplified with PCR before STR analysis can occur. Third type of test that is going to be performed in this investigation is variable number of tandem repeat analysis, also referred to as VNTR analysis. This is what will be performed on my personal cheek cells to rule out my own DNA from the suspect pool. This analysis, repeating regions of 10 to 100 base pairs that are found within the anonymous and polymorphic regions of DNA are analyzed. The patterns of these repeating regions are inherited following Mendelian inheritance patterns from your mom and your dad. Therefore, you can be heterozygous and homozygous at a specific locus, just like what was seen with the STR regions we talked about a few minutes ago. During VNTR analysis, the DNA is first cut with restriction enzymes. These cut in a way that fragments are created, which allows comparison for the number of repeats at a specific locus. To perform VNTR analysis, the DNA cannot be old or degraded, and this is because we often need at least 700 base pairs intact and in order to correctly perform this analysis. It is also common that PCR is also performed prior to analysis to amplify the VNTR sequences. VNTR analysis is very beneficial because the pattern of inheritance seen in each individual is very specific to that individual. However, a limitation is that this analysis cannot be performed on old or degraded DNA, and it cannot be automated. Okay, so I've told you guys about PCR a couple times. It was used in this crime scene investigation on both the VNTR analysis and the STR analysis. So now I'm going to just talk to you guys a little bit about PCR so you guys know what it is going forward. PCR is the polymerase chain reaction. It was created in 1983. It has numerous varieties of application, and it is a standard in many labs today. What PCR does specifically is it copies a specific sequence in the DNA over and over and over and over again, and it amplifies this sequence so that you can get hundreds or thousands of copies of a specific sequence of DNA. There are 4 different steps of PCR reactions: denaturation, annealing, extension, and repeat. During the first step, denaturation, the reaction mixture is heated, allowing the 2 strands of DNA to separate, creating 2 single-stranded pieces of DNA. Both of these pieces of DNA are used as the template strands in the following steps. During annealing, the primers anneal or bind through hybridization to the DNA on the flanking sequences of your DNA that you want to be amplified. These DNA sequences are just actually short oligonucleotides of DNA, and they bind through hybridization. The third step, extension, occurs when Taq polymerase, which is just a heat-resistant DNA polymerase, adds DNTPs along the templates, starting from the primers, to create a new complementary strand of DNA. And the fourth step is just repeat, in which you repeat these steps over and over and over again until you create as many copies of the DNA as you desire. Here are the components of the PCR reaction. These can all be added to the test tube at the beginning of the reaction. Obviously, you need a buffer and the DNA template. You also need the primers and nucleotides to allow extension. Remember, I mentioned earlier the heat-stable polymerase. We use Taq DNA polymerase, which is stable at high temperatures. And you also need magnesium to allow the reaction to proceed. This is just a quick visual to help you guys visualize the PCR reaction. Also, hint, hint, please pay close attention to this figure because there will be something like this on your final exam. And note where I have labeled each step as occurring. Mentioned earlier, PCR was used in both STR and VNTR analysis. This is because there wasn't a large amount of DNA available for both of these tests, so we use PCR to amplify the repeated sequences within the DNA. PCR is quite straightforward to perform in both of these tests because the DNA sequences that flank both the STR and the VNTR repeated regions are almost identical throughout the entire human population, making it easy to use primers and to amplify just the wanted repeating sequences. Okay, class, now that I have given you all a fair amount of background information on each of these tests, I will be going through the results of each analysis as they become available to me. To protect everyone and keep everything confidential, I will not be giving out the identity of any suspect. I will just be referring to them as suspect 1, 2, 3, or 4. Before we move on to actually analyzing the results from this crime scene, I think it's important for me to tell you guys a little bit about gel electrophoresis. Gel electrophoresis is defined as the migration of ions in an electric field. Gel electrophoresis typically separates proteins or DNA based on both size and shape and on charge. In this case, we were using DNA, which all has a negative charge, so DNA is separated directly based on size, with the smaller fragments traveling further along in the gel than the larger fragments. I'm going to just quickly explain the protocol of how you would set up a gel, and then I will show you guys a sample gel, and then we will go into the results and analyzing the results from this crime. To form a gel electrophoresis, you first have to set up your gel correctly. To do this, you first turn the tray basically so that it's 90 degrees from how it will be set up when you actually run your gel. This is so that when you pour in the hot liquid gel, it doesn't leak out and get everywhere. Once you do this, you're going to insert a comb. The comb will create the wells that you are going to put each of your samples into. Once you have your tray set up, you are going to put 0.5 grams of agarose and 50 milliliters of TAE buffer into a small flask. You will then microwave this in about 30-second intervals until you see that all the crystals have disappeared. Once the crystals have disappeared, you're going to let this flask cool. When it is cool enough that you can touch it with your fingers without burning yourself, but still it's fairly warm, you're going to add SYBR Safe to the gel, and then you are going to slowly pour it into your tray. You want to make sure you pour quite slowly so you don't get bubbles appearing as you pour. Then you're just going to let this gel sit for about 45 minutes and solidify. Once the gel has solidified, you will see it is now no longer clear, it's a little murky color. You are going to remove the comb and you are going to take out the tray, turn it 90 degrees and put it back in so it is aligned properly. The wells will sit at the black end of the your whole tray thing, and that is because the DNA is going to go into these wells, and your DNA has a negative charge, so you know it's going to run towards the positive end, which is the red end of your setup. So you want the wells to be closer to the black end than the red end. You're then going to fill the box with running buffer. You're going to use TAE buffer in this case, and you want it to be, there's about 2 millimeters of buffer over top of your gel. Next, you will load your ladder and your samples into each well. This is quite tricky to do, so you're going to want to pipette them in very carefully, ensuring that you are getting the samples directly in the wells that were created in the gel by the comb. You are then going to put a lid on your apparatus, plug in the electrodes, make sure you put the black to the black end and the red to the red end, and you are then going to run it under an electric field of 100 volts. You will be able to slowly see your samples leaving the wells if you look carefully. Once your samples have left the wells, you can turn your voltage up to 120 volts. And then you are going to want to just let this run for like 25 minutes. To make sure it is still running properly, you can look for, there'll be a wall of bubbles that indicates an electric field is running through your gel. And remember that when you go to view this gel, the shorter fragments will travel further through the gel than the longer fragments. Here I have uploaded a sample of what a gel may look like once it is ran and visualized. You can visualize the gel numerous different ways. Often this is done by attaching some type of dye to the proteins or the DNA to allow it to be visualized under UV light. In this sample that I've shown you, you can see in lanes 1 and 2, there is a ladder that has been added. In the ladder, fragments of the DNA are of known sizes. You can see the first one is about 200 kD, while the last one is about 6.5 kD. This shows you that the smaller the fragment, the further it's going to travel through this gel. In this case, the gels will have all started in the wells up at the top hand of the gel, and as the electric field was applied, they will travel towards the positive electrode near the bottom of the gel. The gels that you will see from the experiments done in this crime scene analysis are slightly different, but I just wanted to show you guys this to give you an idea of what a gel will look like. So I think now we are ready to analyze the results that were obtained from this crime scene. We're going to start with the RFLP analysis that was performed on the droplets of blood found at the crime scene. Before I display the results of the RFLP analysis on the screen, I just want to mention that you guys should remember the DNA taken from the crime scene and also the 4 suspect DNA was first digested with EcoR1 and PST1 restriction endonucleases, and then a loading dye was added to this DNA before it was put in the wells to allow for visualization of the bands on the gel. The latter did not need a loading dye added to it because ladders typically come pre-made with loading dye already in them. Here are the results that we received from the RFLP analysis. I have labeled each column so you can see what each column corresponds to. The very first column on the far left, labeled with an L, corresponds to the ladder. The ladder used in this analysis was a Hind3 digest of phage lambda DNA. When you digest phage lambda DNA with Hind3, you will get 8 fragments, all of known sizes, and these allow you to create a standard curve and determine the sizes of the fragments in your other columns. The second column is labeled CS. It contains the DNA collected from the crime scene, and the following columns labeled S1, S2, S3, and S4 just correspond to suspect 1, 2, 3, and 4. By quickly looking at this gel, you can see that the genetic fingerprint of suspect 3 appears to match that of the DNA found at the crime scene. You can see that the bands appear at the exact same positions within the gel. You can also see that it is likely suspect 1, 2, and 4 did not commit the crime because their genetic fingerprints do not match that found at the crime scene, meaning their restriction enzyme sites are found at different locations throughout the genome than the DNA that was found at the crime scene. To ensure that suspect 3's DNA is in fact the same DNA found at the crime scene, we are going to go through and calculate the fragment size for each of the unknown bands seen in this gel. To do this, we are first going to establish a standard using the ladder seen in the first column of the gel. To do this, we first measure the distance in millimeters that each fragment of the ladder traveled through the gel. I did this using the superimposed ruler indicated by a red arrow on the right-hand side of the gel photograph. Since the fragment size of each band in the ladder is known, once you calculate the distance each band traveled, you can then create a plot of the log of size of the DNA fragment versus the distance traveled, and use this graph to estimate the unknown fragment sizes. So here we have established the standard curve for this gel, and what you can see is that there is a logarithmic relationship between the molecular mass and the distance traveled by each band. You can now use the equation of the line generated from this standard curve to determine the fragment size of each of the unknown fragments visualized on the gel. I am including one sample calculation for fragment size so you guys can see how this is done. Here I have just calculated the size of the first band seen in the crime scene DNA. As you guys can see, I have used the equation of a line generated from the standard curve created in the plot I just showed you all. I will not be showing you the calculations for all the fragment sizes, but I would like you guys to please pause this video here and take a moment to roughly estimate how many base pairs each fragment seen on the agarose gel would be. So here I have just put on the fragment size calculated for each band from the lab. I'm hoping that you guys did pause this video and calculated roughly the same values. By calculating the fragment size, it becomes fairly apparent that suspect 3's DNA directly matches the DNA found at the crime scene with respect to location of restriction enzyme sites. This is because of the bands seen on both of these DNA, the fragment sizes are calculated to be completely identical, indicating that suspect 3 may likely have committed this crime. The test we are going to look at is the analysis of STR, also referred to as short tandem repeat analysis. Remember that this was performed at the TH01 locus on both the crime scene DNA and all 4 suspect DNA. Remember, this locus consists of the bases TCAT repeating a variable number of times. It's also important to note that this number of repeats varies between individuals in the population, and this variation is referred to as different alleles. Before we dive into analyzing these results, I want to remind you guys of something we talked about earlier in the term. Remember that for a particular given trait, it is possible for an individual to be heterozygous or homozygous. That means it is possible for the individual to have 2 differing alleles on the individual chromosomes of a homologous pair. Therefore, on one chromosome, an individual may have 3 repeats, or on another chromosome, the individual may have 7 repeats. This would mean that the individual has a 3-7 genotype with respect to the TH01 locus. If the individual had 7 repeats on both chromosomes, they would just have a 7-7 genotype with respect to the TH01 locus, or just a 7 genotype. So here I've uploaded the results from the STR analysis for you guys to visualize. As you can see, once again, I labeled each column, L for ladder, then crime scene, suspect 1, suspect 2, suspect 3, and suspect 4. Remember that this test was performed on the DNA found from the skin cells that were left on the murder weapon, and that before this test was run, the DNA was amplified using PCR at the TH01 locus. Once the PCR reaction was completed for both the crime scene DNA and each of the 4 suspect DNA, an orange G loading dye was added to each of the mixtures to allow for visualization on the gel. A ladder was also added to the first column of the gel. In this STR analysis, the ladder used was simulation TH01 allele ladder. In this ladder, each of the specific bands seen correspond to a specific size of one of the TH01 alleles. The number of repeats corresponding to each fragment of the ladder, I have simply put to the left-hand side of each ladder fragment. So as you can see, the smallest fragment contains 3 repeats, where the largest fragment contains 10 repeats. And as you can see, once again, the smallest fragment containing only 3 repeats migrated the furthest, while the largest fragment migrated the slowest. So here I have analyzed the crime scene DNA at the TH01 locus. This was simply done by looking at where the fragments lay from the crime scene DNA and comparing this back to the ladder to determine the number of repeats seen at the TH01 locus. Immediately, you can see that the DNA found at the crime scene was heterozygous because 2 bands are seen. This means that on each chromosome of the homologous pair, there is a different number of repeats at the TH01 locus. By comparing the DNA at the crime scene to the ladder, it is apparent that the DNA found at the crime scene has a 5-8 genotype at the TH01 locus. Now would be a great time for you guys to pause this video once again and practice analyzing STR results. I would like you guys to please analyze the STR alleles seen in each of the suspects' DNA. So here are the TH01 genotypes for the rest of the DNA samples. I hope you guys all got close to this when you tried it on your own. From this, you can see that suspect 3 once again clearly matches the DNA profile seen at the crime scene. Both suspect 3 and the crime scene DNA are heterozygous at the TH01 locus, and one chromosome contains 8 repeats while the other chromosome contains 5 repeats. The DNA collected from suspect 1, 2, and 4 can be determined to be different from the DNA found on the murder weapon at the crime scene because the number of repeats at the TH01 locus is different from that found at the crime scene. The third and final test that was performed in this crime scene investigation was VNTR analysis, or variable number of tandem repeat analysis. And this test was performed on my own DNA along with my partner's DNA to eliminate ourselves from the suspect pool. I know we talked about how to properly perform a cheek swab earlier in the term, but I'm going to give you guys a little hint, and you will have a question on this on your final exam in a couple weeks. So I am going to review the steps one more time right now for you guys. First, what you're going to want to do is you're going to want to lightly chew the inside of your cheek, just enough to get some loose cells in there. You're then going to want to rinse your mouth very vigorously for about a minute with saline solution. You only need about 10 milliliters of this solution. Once you have rinsed your mouth fully, you can spit the saline solution into a cup, and then you need to swirl the cup to gently resuspend the cells, and then transfer about 1.5 milliliters of this solution into a top screw microcentrifuge tube. After this, you are going to spin the tube on the microcentrifuge for approximately 2 minutes at 13,000 RPM. When you are finished spinning this, you guys need to check that your buffer is clear, not cloudy, and that you have a visible white pellet at the bottom of your tube. If your solution is still cloudy, spin the centrifuge tube one more time. If your solution is clear and no pellet is visible, that means you likely need to re-chew your cheek and get the saline solution, rinse out your mouth. Basically just repeat, because you obviously did not get enough cells. So what you're going to do is, once you get a pellet about the size of a match head, you're going to pour off the supernatant, basically just the saline solution, and you're going to be very careful to keep the pellet in the bottom of the centrifuge tube. Once you've done this, you're going to repeat once again steps of gently swirling your cup that should be about 10 to 15 milliliters of saline solution with your cheek cells, and then you're going to repipette 1.5 milliliters into the microfuge tube, resuspend, and you're going to do this approximately 2 more times just to make sure you get a big enough pellet. That just means you want to make sure you have enough DNA cells to properly do this analysis. Once you have collected just your isolated cells, you're going to resuspend these cheek cells in about 140 microliters of the lysis buffer by using a pipette to mix or by vortexing vigorously. You are then going to cap this tube and place it in a water bath at 55 degrees Celsius for approximately 15 minutes, and once that 15 minutes is up, you will quickly vortex the sample to remix it. You then need to incubate this sample on a 100 degree Celsius heating block for 15 minutes. Following this, you will remove the tube, allow it to cool for 2 minutes, then vortex for approximately 10 seconds or mix by slightly tapping the edge of the tube. Following this, you will spin the tube in a microcentrifuge for 2 minutes at 6,000 RPM. This is going to pellet the cell debris and leave the DNA in the supernatant. So this is opposite as before when our DNA was in the pellet. Now our DNA is going to leave the pellet and be in the liquid top part of the solution. Once you have your DNA in the supernatant, you are going to, and I cannot stress this enough, very carefully transfer approximately 80 microliters of the supernatant to a clean microtube. And you need to do this super carefully because you do not want to disturb the pellet at the bottom of your sample after centrifugation. This is because if even a couple of the Chilex beads are transferred with the supernatant, they may take all the magnesium that is required by Taq polymerase during PCR, and PCR reaction will not happen, meaning your DNA will not be amplified, and when you go to look at the results, you will have no results. Once you very carefully transferred over this supernatant to a clean microtube, you're going to sit this on ice, and then you are going to set up a PCR pellet tube for PCR. You're going to do this by placing a PCR reaction bead in the tube and slightly tapping the tube to ensure this bead goes all the way to the bottom. You are then going to add D1S880 master mix and your DNA solution to this tube, and then you are going to gently mix the tube by inversion. This just makes sure that all the reagents are at the bottom of the tube and they aren't sitting at the top. Once the bead is completely dissolved, you're going to keep the reaction on ice until it is ready to go into the PCR cycler. Once you put it in, the PCR reaction will take about 3 hours approximately to complete, and then your DNA can be put on a gel, and your results can be analyzed. The steps were carried out exactly how I just mentioned to you guys on both mine and my partner's cheek cells, and our DNA was analyzed at the D1S80 locus to eliminate ourselves from the suspect pool. VNTR analysis is carried out in almost the same manner as RFLP analysis, in that the latter is first used to create a standard curve, and then the equation of the line from the standard curve is used to determine the fragment sizes of the unknown bands in the gel. In VNTR analysis, once the fragment size of each band seen in the gel is determined in base pairs, the number of repeats seen at each allele can then be determined by looking at a table which gives you the allele sizes and frequencies seen at the D1S80 locus, such as the table I'm showing you guys here. For example, looking at the top row in this table, you can see that if the allele size of an unknown fragment was calculated to be approximately 369 base pairs, this would mean that on that particular chromosome, the person has 14 repeats at the D1S80 locus. For this analysis, the crime scene DNA was ran on a different gel than mine and my partner's DNA, and because of this, two different standard curves needed to be created, one for the gel with the crime scene DNA and one for the gel with mine and my partner's DNA. This is because each gel is slightly different and you need to create a standard curve for every individual gel you analyze. Because of this, I am going to analyze the crime scene DNA first, and then I will analyze mine and my partner's DNA, and then we will compare what genotypes are seen and determine if it was possible that me or my partner could have been committing this crime. So here is the gel that corresponds to the VNTR PCR analysis for the crime scene DNA. Once again, I have labeled the columns L for ladder and CS for crime scene. On the ladder, I have also went through and indicated the size of each fragment. Since the EDVO-Quick DNA ladder was used, the band sizes in base pairs were known for each fragment that appears on the ladder. Typically, a 200 base pair fragment shows up, although it did not appear in this gel. Using the same techniques seen in RFLP analysis, the distance that each band in the ladder migrated was calculated in millimeters, and then a graph of log band size versus distance traveled in millimeters was created. Once again, the equation of a line created from this graph was used to determine the fragment size of the unknown bands within the agarose gel. By using this method, the two fragments seen in the crime scene DNA were determined to be 398 base pairs and 620 base pairs. This corresponds to a 1630 genotype at the D1S80 locus, indicating that the individual who committed this crime is heterozygous at the D1S80 locus. Here are the results for both my own and my partner's VNTR DNA analysis at the D1S80 locus. Once both mine and my partner's DNA was done undergoing the PCR reaction, a blue loading dye was added to each sample to allow for visualization of the bands in the gel under a UV light. Looking quickly at the gel, it is apparent that both me and my partner are heterozygous at the D1S80 locus because only one band is seen in our fingerprint. Once again, the EDVO-Quick DNA ladder was used. This column was labeled with an L. The M column indicates my personal DNA, where the P column indicates partner's DNA. Since the EDVO-Quick DNA ladder was used, fragment size of each band in the ladder column was known, and the distance traveled by each band in millimeters was able to be calculated and then used to create a standard curve in order to determine fragment size of both the unknown bands in mine and my partner's DNA. Once the standard curve was created, once again, the equation of the line from the standard curve was used to determine the fragment size of the unknown bands. Using the standard curve for this gel, it was determined that both me and my partner were homozygous with 19 repeats on both chromosomes at the D1S80 locus. Since the crime scene DNA was found to be heterozygous with a 1630 genotype at the D1S80 locus, it is apparent that we are able to eliminate both myself and my partner from the suspect pool, since our DNA differs from the crime scene DNA at the D1S80 locus with respect to the number of repeats seen here. I want to talk about what the findings of these results mean with respect to who committed the crime. I do just want to touch on a few important subjects that you guys do need to know for the final exam, and these subjects are very, very important in forensics analysis. They were used in this crime scene investigation, although I didn't feel like getting into detail about the results and the findings. So instead, I am just going to briefly touch on the concepts so you guys have an idea about what these are. First, I want to mention the CODIS system, which is the Combined DNA Index System. This system is a computer program that collects a database of DNA from either convicted or suspected criminals from the US, Canada, and also from about a dozen other countries. What this system does is it specifically looks at 13 different STR loci that are uniformly distributed throughout the whole human genome. And each one of these loci are from an anonymous region, meaning that they will not reveal any personal medical or health information about the individual, and that just helps keep the confidentiality of the individuals intact. The CODIS system looks at 13 different STR loci, which are distributed evenly across the human genome. The photo I am showing you guys here shows the positions of all the different STR loci that is looked at within the CODIS system. The CODIS system chooses to look at 13 loci because this increases the ability to distinguish between two different people based solely on their genetic profile. And it also decreases the probability that a match occurred solely by chance. This makes sense because it is highly likely that I am identical to someone else with respect to just one locus. But if we were to look at, say, 10 different loci, it is much less likely that I am genetically identical to someone else at all 10 of these loci. Another gene that is typically used in crime scene investigations is the amylogenin gene. This gene is very important because it gives information about the gender information of a particular DNA sample. This is because the amylogenin gene is present on human sex chromosomes, that is the X and Y chromosome, and the pre-CR products of the amylogenin gene produce both X and Y chromosome-specific products that are of different sizes, and these can be used to determine whether the DNA sample is from a male or a female. Another very important calculation performed in forensic analysis is the RMP calculation, or the random match probability calculation. This calculation takes a few different factors into consideration, including the allelic frequencies seen throughout a population, the Hardy-Weinberg equilibrium, which assumes that the distribution of alleles within a population are in Hardy-Weinberg equilibrium, and the product rule, which just states that the total RMP can be determined by multiplying the calculated RMP for each specific locus together. The calculation is very important because it tells you the probability of a match occurring just by sheer chance. For this crime scene investigation, the RMP was calculated for the suspect 3 at the TH01 locus to determine the probability of the match seen between suspect 3 and the crime scene DNA occurring by chance. When STR analysis was performed at the TH01 locus, it was seen that both suspect 3's DNA and the DNA found at the crime scene were of a 5-8 genotype at the TH01 locus. Since the individual is known to be heterozygous at this particular locus, and the number of repeats is known for each chromosome, the allele frequency can be simply looked up for the number of repeats seen on each chromosome, and from there RMP can be calculated. Here I have included a sample RMP calculation for suspect 3 at the TH01 locus. This is simply determining the probability that suspect 3 matched the crime scene DNA exactly at the TH01 locus. As you can see, you simply use the heterozygote locus RMP formula 2PQ to determine RMP. As calculated here, it is apparent that there is a very low probability of this match occurring by chance. However, analyzing more than one locus would ensure that this chance decreases. Before I go over the results seen from this crime scene analysis, I would like you guys to please take a moment, pause this video, reflect on what you've learned, what we've brushed up on, and take a moment to see if you can determine which suspect committed this crime based solely on DNA evidence. Seen from both the restriction fragment length polymorphism analysis and short tandem repeat analysis indicate that the genetic fingerprint seen of suspect 3 directly matches the genetic fingerprint produced by the DNA collected from the crime scene. When RMP was calculated for STR at the TH01 locus, it was found to be 0.004769. This indicates that there is a very low probability this match occurred by chance. As a result of this DNA evidence, suspect 3 has been taken into custody by the Kelowna Police Department and is currently awaiting trial. Guys, you did it. You made it to the end of the semester. I know this was a bit of a heavy topic to end on, but I thought this was a really unique way for you guys to see how DNA analysis has become an essential tool in the criminal justice system for identifying possible criminals. I also thought this was a really unique way to tie together different concepts we have been learning about all term. If you guys have any questions or concerns before the final exam, feel free to email me, and if not, I will see you there. Good luck!